Good evening, everybody. Is this on? You hear me? Good evening. Hello. Is this on? The light's on. Where is? Right now, volume is up. Oh. Let's try it. Okay, we'll get started and we'll get the sound sorted. So, good evening, Provost, Vice Provost, ladies and gentlemen, colleagues, guests. You're all very welcome. And on behalf of the Faculty of Health Sciences, it gives me great pleasure to introduce Professor Aideen Long, who will shortly present her inaugural lecture. The inaugural lecture is an important recognition event that celebrates the appointment of full professors in Trinity College. And Professor Long was promoted to Professor of Cellular and Molecular Immunology in 2023, and that's why we're here this evening. And I'd also like to welcome all Aideen's friends and family, her parents, sister, brother, daughter. I think one is joining us by video link as well. And you're all very welcome as well to celebrate with us this evening. Now, more than most professors in this situation, Aideen has shared very little of the person and the personality behind the successful scientist in her bio, being naturally private and self-effacing. A colleague remarked to me that Aideen is a very private person. She's self-deprecating, does not like the spotlight on herself. So it's my job to ignore that completely, tell you about the real Aideen Long. So Professor Long is not shock horror of Trinity, but luckily for us, she found her way here twice and stayed. Raised in Wexford, a GP friend of the family advised Aideen to study biochemistry and then go on to research immunology, which was not the mainstream pathway that it is now. So whether that was intuition or insight, I'm not sure, but Aideen enjoyed science and found this was indeed a pathway that served her well. She completed her primary degree in UCD and then did her PhD with Owen Ward in DCU, who complicated matters by moving to the University of Waterloo in Canada, where Aideen followed for her doctoral research. And I only found out last week that while Aideen was doing her PhD in Ontario, I was 3 hours down the road in Rochester, New York, doing my PhD. At any rate, the time in Canada was allowed Aideen the opportunity to travel and see some of the country with her sister, while Peter, I understand, was an occasional visitor. So having successfully completed the PhD, it was time for Aideen to return to Ireland. Being a big football fan, make sure that her honeymoon included Italia 90, before the real start of her career in immunology. And her first mentor, Dermot Kelleher, told me that Aideen was my first postdoctoral fellow and the cornerstone of the development of immune signaling research and translation at the Sir Patrick Dunn's Laboratory, and then the Institute for Molecular Medicine at the TCD St. James's campus. This was a new field for Aideen, but she rapidly built an international reputation in the study of intracellular signaling in T lymphocytes and how this could be analyzed and modified from a disease-related perspective and thence a translational perspective. Her energy and drive were pivotal to the development of the Institute of Molecular Medicine, now Translational Medicine, and her mentoring of younger scientists was exemplary, an outstanding scientist and an outstanding person. So Professor Long obviously went from strength to strength, building a stellar reputation in immunology and cell biology. She was the first in Ireland to receive a Wellcome Trust Career Development Award, and after a spell in RCSI, returned to Trinity as a senior lecturer and principal investigator. Aideen characterized her work as transformational, finding solutions including diagnostics, therapeutics, and innovative approaches like cell therapy. It's an integrative triangle of basic, translational, and innovative research. Her work has significantly contributed to our understanding that cell migration is the basis of inflammation and cancer metastasis, but she also co-leads a landmark translational project where clinicians and scientists are working together to design testing modalities, vaccines, and therapeutics for COVID-19. including her own interest and how the immune system responds to SARS-CoV-2. So Aideen has many strings to her bow, but her special expertise in T-cell characterization and migration has led to several industry collaborations where basic biological mechanisms are exploited for innovative treatment of cancer and inflammatory diseases. This is not only very successful for Professor Long from an intellectual point of view, but it has also brought in tens of millions of euro in grant funding, publications in top journals, prestigious lectures, and international recognition. Over the years, a strong and tightly knit research community developed here on the hospital campus, though our Vice Provost, Orla, confirmed that it was not all work and no play, where Aideen was often the instigator of practical jokes on the unsuspecting new members of the lab. I have some examples, but maybe we'll talk about it later. Of course, science at this level is a team sport, and Aideen, being captain of the team, has a long history of training and mentorship of students and emerging researchers. Our very first PhD student, Michael Freely, is now a staff member in DCU and still a collaborator. Michael's not here, is he this evening? No, but I spoke to him the other day and he said that once he decided to work for Aideen, it was a decision I've never regretted. I've met many great people in science, but I can honestly say that Aideen has always been top of the list. It goes without saying that she's super intelligent, has unwavering dedication to uncovering the complexities of immune cells and cancer cells, is ambitious, super educator, and endless energy. But she somehow combines this with compassion for her staff, students, and colleagues alike, is always there to lend an ear, and her door is always open. She is there to solve any crisis, and she has this knack of putting the positive in the negative. And that's something I heard over and over again about Aideen: her warmth, her generosity, her kindness, and her loyalty. And I think that's admirable that these are the traits that friends, colleagues, and students highlight about Aideen, equal to her scientific and professional achievements. By the way, one of which legacy is the outstanding MSc in biomedical science, which Aideen developed and directed. But we all know there's no advancement in Trinity without service to college and discipline. For Professor Long, it's been consistent with her ethos of service and willingness to help others. She took on what we would call a chunky job of Dean of Graduate Studies, where she has had a reputation of fairness, hard work, and prioritizing the needs of students, going as far as posing for a charity calendar for students, which I understand celebrated nature in its the most tasteful way. You can ask the Dean of Students about that. On then to her current position as Director of TTMI and weathering the challenges of COVID, while contributing to our knowledge and preparedness for this and future pandemics. It's no exaggeration to say that Professor Aideen Long is an example and a significant contributor to the success story of Irish science over recent decades. But apart from the hard work, integrity, and dedication to your team, I understand you're sports mad and not averse to the odd party. So whatever your secret is, Aideen, I hope you keep it up, and I welcome you now to deliver your lecture, From Biotechnology to Immunology, a journey of a cell biologist under the virus.
Well, where do I go from there? Thank you very much for that very kind introduction, Brian. So I guess maybe start at the beginning. I was born in Dublin and Dad was working in the bank, so got transferred from place to place. So quite young, we moved to a small village in County Wicklow called Tinnahealy. And we lived in this house here. And thankfully, we actually didn't live there when this happened. This is when some unsuspecting thieves got a JCB to try and take the ATM out of the front wall and took the whole front off the house. But as I say, we weren't there at the time. We moved to Enniscorthy, and I probably spent most of my childhood in Enniscorthy because I went to primary school and all of my secondary education was in Enniscorthy, and then the folks moved on to Rosslare. And as Brian said, I went to the opposite end, or the opposite camp, UCD to study biochemistry. And when I finished my degree in biochemistry, I didn't really know what I wanted to do, but I ended up going to DCU to work on a project, and I actually had to reach out my PhD thesis at the weekend to find what it was about. The title being The Production of Ace-Loin Compounds in Saccharomyces cerevisiae by Biotransformation. And that's where my career in biotechnology started and finished, really, because I started in DCU. Saccharomyces cerevisiae is baker's yeast. I used to pick up baker's yeast in the Irish Yeast Company, which many of you will be familiar with. It's down by the side of Trinity on College Street, and bring it to the lab and weigh it out and do the experiments. And I didn't fall in love with this particular field of research. It had a lot of biochemistry, but it just wasn't for me. And I was thinking, well, when you finish your PhD, what am I going to do next? So as it happened, I bumped into a mentor from chemistry department in DCU when I came back from Canada and finished my PhD. And he had a copy of Nature in his hand, and he showed me a small little lad which said wanted a postdoc to work on protein kinase C in T cells in Trinity. I looked at it and I thought, I didn't even know what protein kinase C, even though I'm a biochemist, I had to look up what protein kinase C was in a textbook. And at the time knew nothing about T cells, but I applied anyway. And lo and behold, went for an interview in December. I didn't cover myself in glory, it has to be said, with that kind of background. But anyway, I was offered the position and ended up with Dermot Kelleher. And this building actually wasn't here at the time. So I was over in the Sir Patrick Dunn's labs over in the pathology laboratory. And this was the medical school. So what is this big, nice building now was that prefab over where the new IBTS is now. And this was like quite a smelly, fox-ridden building at the time. But anyway, started off working with Dermot and very quickly got into working on protein kinase C and T cells, and really got to like this protein kinase C and T cell area. So the research flourished at the time, and more people came along to work with us. And we started to publish. This is the first paper we published. And then we started to publish more papers in the at the time. But actually, this will bring I hope, a lot of memories back to some of the people in the room. The Sir Patrick Dunn's labs, and at the time I started, it was just myself and Anne Murphy. As Brian said, Dermot's first postdoc, Dermot here. I actually did some work in the lab, sometimes with a white coat on, sometimes not, which is like a total no-no. And gradually the group expanded and flourished. And you probably know or recognize some of the people here. We have Barbara Ryan, who's now at Tala, Susan McKiernan, who is a consultant herpetologist here now, Henry here lurking in behind, Rachel Doyle, Dearberny, Ein, and Yuri joined the group. And you know, we have to remember people like Yuri, Nasser, Francis, no longer with us, but really great part of the gang at the time and the work that was going on. Ross here, and a PhD student of Ross, Jackie Daly. And, you know, while the research was great and the work was great, the fun was really great. You know, and there was lots of fancy dress parties. This is Alex Whelan, for those of you who don't recognize him. And Richard Farrell, who's I think out in Blanchardstown now. Other people that I couldn't find photographs of were in the lab at the time, including Orla, Joe McPartin, I see up there, Mary O'Sullivan. So lots of the old gang, you know, in a time when we were in an older building, but having great fun doing research. Then, as Brian says, I went to the RCSI, and my first PhD student, Michael Freely, who also then took up the mantle of working on T cells, and particularly protein kinase C in T cells. So we had to keep that going. But while I was in the RCSI, I got involved in other projects and apart from T cells, so other cell biology projects, which were great. You know, I shared a PhD student, Irish born, with David Probe, and we looked at a protein actually that's normally only expressed in T cells and white blood cells, but is expressed in colon cancer cells, and there it can do some damage. So by looking at other cells and the biology of other cells, I was learning an awful lot about cell biology in general and T cell biology in particular. So then in 2005, I came back to Trinity, and what this building now was the Institute of Molecular Medicine, now what we call the Trinity Translational Medicine Institute. And that move, I guess, was the start of, you know, a huge adventure in Trinity in terms of not just the research, but, you know, taking part in college life and doing jobs within the university. So for 2013, 2016, as Brian says, I was Dean of Graduate Studies and very lucky to work with two incredible people in graduate studies. It was a tough job, but it was a brilliant way to meet other people, to see what, you know, the graduate student cohort were up to across all the faculties, and so importantly, to meet other academics from other faculties. And I had the great pleasure of working with people like Barry McMahon in the Innovation Academy and others during that time as being Dean of Graduate Studies. Then I took over as Director of TTMI in 2019, and that was just before COVID. So we headed into quite a tough time in terms of the university and research in general. But from our point of view, it signaled the initiation of a great collaboration between another institute in the university, Trinity Biomedical Sciences Institute down in Pier Street, and our colleagues here in St. James's, our clinical colleagues, and a really great project where we were looking at different aspects of COVID-19 on a very collaborative basis, clinicians and scientists working together to try and solve problems associated with COVID-19. And when Orla was Dean, she started the UNICOV project, and we took up the mantle here in TTMI and led the screening of students for COVID-19 at the time, and particularly focusing on disease, it doesn't matter, when the symptoms weren't visible. Asymptomatic disease, exactly, thank you. So just trying to keep the campus safe and get the students back safely. Then in what we might call the post-COVID times, one of the highlights of being involved here in TTMI was leading out on the launch of the Translational Medicine Alliance Ireland. So this is the network of 10 universities doing translational medicine research, working together to try and improve the visibility, the quality, and everything we do in terms of translational medicine nationally, so as we can be more international facing. So coming back, I brought back, managing to get some money from RCSI, and on the way back, I brought back a research team and we got going. And, you know, from a point of view of cell biology, one of the things that I focused on, when I came back was in esophageal cell biology. So the cells that line your esophagus, we looked at the effect of bile acids and how bile acids can promote inflammation and ultimately sometimes cancer in these cells. And these cells here are migrating esophageal cells. And you'll see when we come to talk about migrating T cells, how alike they actually look. So they have very many common mechanisms, even though the differences can be quite striking as well. But for the purposes of this evening, I want to focus the scientific part of the talk on the T cell. So going back to where I started when I came to Trinity, I suppose what I would call my research love, and tell you a little bit about some of the research that we have been doing and continue to do in the area of T cell biology. And I hear you all saying, what are T cells? Very excitedly. Well, T cells are actually white blood cells that are, I would say, the commanders of our immune system. If we were to just very briefly say what T cells do, they recognize and they remember pathogens. They release chemical messengers that we call cytokines, and these orchestrate or direct the immune system or the immune response. They can recruit and instruct killer cells, and they are themselves killer cells. So this is a picture here of a T cell killing a virally infected cell. So T cells can physically kill virally infected cells, and they do that by literally injecting chemical poisonous toxins into the virally infected cells and killing them. They can also direct or orchestrate these cells to make antibodies. So already you can see that T cells are very powerful cells. And when we talk about powerful cells, we talk about cells that can protect us, but also cells that if they're not tightly regulated, they can cause damage because of the powers that they have. And that's what happens when we have T cell mediated inflammatory diseases. So in these diseases, they are, and you'll have heard of all of them, they are caused by centrally or at their core T cells. So in the case of psoriasis, T cells make their way into the skin, and by doing the things that I've just described to you, they can cause tissue damage and inflammation, that is psoriasis. In multiple sclerosis, T cells can make their way into the brain, and using the powers that I've just described to you can cause damage in the brain to cause this debilitating disease called multiple sclerosis. And in the gut, this is what the inside of a healthy colon looks like. If you have inflammatory bowel disease that includes ulcerative colitis and Crohn's disease, you get these inflammatory lesions in the gut mediated by T cells. So T cells go in there and they do their thing and cause this swelling and inflammation. So the key is that T cells are important to protect us, but if they're not tightly regulated, they can do us damage. And this here is the kind of central model of this inflammatory response or T cell migration. So this is basically how T cells get into the tissue. So in the normal situation, we know that our white blood cells, including our T cells, are flowing around our bodies in our bloodstream or in our circulatory system. But if they come across in the blood vessel close to a tissue where there is injury or infection, the blood vessel becomes sticky. The T cells, first of all, they start to roll along the blood vessel, then they stick to the blood vessel, and then they change shape, they crawl along the blood vessel, and they squeeze in between the cells and into the site of inflammation. And this is what we want to happen. We want this to happen so long as this tissue inflammation resolves itself when the damage is cleared or the pathogen or infection is cleared. Let this resolve. And it's when this isn't resolved, we get those inflammatory diseases that I just mentioned. We get this redness and swelling and inflammatory disease that I've just described. And in the lab, we can recapitulate this step here. So this is where we coat plastic surfaces with proteins that mimic or simulate the blood vessel. And the cells stick and they change shape and they crawl around looking for places where they can actually squeeze into the blood vessel. So these are T cells that are trying to get in. So we're simulating this step here where the cells polarize, crawl and migrate into the tissue, causing inflammation. And another area where we want T cells to come in and do their thing is in a tumor. So you want T cells to be able to come into a tumor site, be inflammatory, and kill tumor cells. So the key is to control it. You'll hear me then mentioning during my slides, we stimulate cells to migrate. And it's this kind of model that I'm talking about. We put these cells on a surface that mimics blood vessel and they crawl around like that. We can actually go a step further and try and simulate what would happen in normal situations by coating little channels with blood vessel cells. So cells that line our blood vessels are called endothelial cells. We could line little channels with those endothelial cells, put in T cells under flow and see what happens. So if there's no inflammation, you can see that the cells are just scooting on by and they are flowing through like they would in the normal blood vessels. However, if we put in an inflammatory like signal, you can see that the cells are sticking to the cells like they are going to actually move into the tissue. And if we look at this quite closely, you can see that that's actually what happens. So this cell is mooching around, as are some cells here, and eventually crawls in and disappears into the tissue. So we can study how this inflammation might happen in the body, in the lab, using these models. Right, let's go back to the schematic diagrams for a minute, because I want to just point out the idea that every cell has a skeleton. And this is very important because if cells are going to move like they do in this crawling along blood vessels and into the tissue, they have to have a skeleton, right? So that's what the skeleton looks like schematically if the cell is resting. So if the cell is just flowing around in the circulation. And there are two important parts of this skeleton that I'll be referring to. Actin, which is here in red, and microtubules in green. So when the cell sticks to the side of the blood vessel, it starts to polarize and form that tadpole-like cell, if you like, that's able to crawl along the endothelium. And the important thing is here that the actin, which is the red one, that's like a scaffold. So actin acts like a scaffold at the front and a bit at the back of the cell, and it acts to push the cell forward, right? And the microtubules, which are these long green train track-like structures, they also, they polymerize, or they get bigger as the cell moves forward, and they depolymerize or they decrease as the tail is pulling behind. And another important thing that these train-like structures do is that they act a bit like pulleys or production lines. They carry proteins from the back of the cell to the front of the cell as the proteins are needed to drive the cell forward. And this is what a very high magnification microscopic image of a T cell looks like in this state. And then just to remind you, for us to change shape, you need a skeleton. For the cell to change shape, you also need a skeleton. So how do we study? We want to be able to study scientifically this process of T cell migration. Well, we can look at the videos, which are, cool and interesting, and they tell us something, definitely. And you can see that the cells are moving around at a certain speed, but we can actually make measurements of those. So we can tell how fast the cell is moving. We can track them. We could tell if they're moving all the time in the same direction, or if they decide to not move in one direction and move in another. So we can, by just even analyzing videos, we can tell a lot about a T cell and changes then that happen if we treat it with a drug or treat it with something that might prevent that. We can simulate the idea that a cell will move from the circulation into the tissue by coating a well with basically the endothelium or the cells of the blood vessel or proteins that might emulate that, and count how many cells go through. So we can look at cells in, or we can take these cells, fix them so they don't move anymore, and stain them and look at proteins in the cell that are involved in migration. And in looking at this image, this is actin here and here at the back, and this is microtubules, the train tracks that I was talking about. But we can look at other proteins and see where they are relative to these, where they are when the cell is resting, where they are when the cell is migrating, et cetera, et cetera. So it can tell us a lot about the actual mechanism of cell migration. And then if we treat this with a drug, we can see all sorts of things happening. So the cell will round up, or the cell will form these stars, or the cell will have long tails, et cetera, et cetera. So looking at the cell under the microscope and staining for different proteins can tell us an awful lot about what's going on. So I'm just going to tell you a few very short little stories about some of the research that we've done in this. And the first protein we looked at in this context is a protein called RAC1. And that is really immaterial. Safe to say that RAC1 is a protein that's like a wheel, and we call it like an adapter protein. And just like any adapter, it basically holds things together that might be important in any kind of a reaction or any kind of a process within the cell. So we figured that RAC1 might be important in this process of T cell migration. So we looked at where, first of all, is this protein in the migrating cell. And if you look here, this is RAC1 here, at the front and at the back. And it actually looks like where actin would normally be. But when we look at the two of them together, we could see that they're actually not co-localizing very much. Because if you see two proteins, one is stained red and the other is stained green, and they co-localize, you'll see yellow. But in this case, we still mainly see red and green and just a tiny little bit of yellow here. So on first inspection, things can be deceiving, and actin actually doesn't have much of a relationship with RAC1. However, when we looked at the tubulin part of the skeleton, the long train tracks, what we found was that there was a lot of co-localization, for want of a better word. So the length of the tracks, there wasn't much overlap, but at the tips of the tracks, both in the resting and in the migrating cell, it's the tips that are co-localizing with the RAC1 and microtubules. And this is actually very important, because when we looked then and analyzed cells that we knocked out RAC1 expression, we took T cells and we knocked out RAC1 expression, they didn't have it, and we compared it to cells that did have RAC1, what we found was that the cells that didn't have the RAC moved more slowly, and they didn't move in one single direction. So they lost this ability to move one single direction. And when we actually focused in on that, so this is the normal cell, you can see the usual shape like a tadpole, moving forward, little tail, and it's moving around. The cells in which we had knocked this protein out couldn't decide which way they wanted to move. So they were basically moving in every which way direction. So they were moving this way and that way, and this way and that way, and explaining why they actually couldn't move forward in a single direction. What we showed was that this protein was very important for this treadmilling of this long microtubule skeleton. And that's why these cells couldn't actually move or commit to moving in any direction particularly. So that's RAC1. So the next little story is about two proteins, RAB5 and PKC epsilon. Moving back to PKC, one of the PKCs. So this is a migrating cell, and just to confuse you, but this is the microtubules. Usually I show it to you in green, but this is it in red. So these are train tracks here, shown in red here, because we have stained this protein called RAB5 in green. And RAB5 is basically a protein that coats these little capsules. And these little capsules are bringing proteins back and forth in the cell along the microtubules, so along the train tracks. So these little capsules literally treadmill along these train tracks and bring proteins up and down the cell as it needs to move. So what we actually found was that if we looked at a cell that was stimulated to migrate and shone a laser on it to bring up the little capsules, we found that it actually associated or interacted fleetingly with this enzyme PKC. And this was a very meaningful interaction. So it had to happen. We showed that PKC, that this protein was a substrate, not to get too complicated about it, but this protein was a substrate of this enzyme. So they had to interact for the reaction to happen. And you can see, you know, this just in still pictures here. So this is what happens if we shine a laser on the normal cell. So in the normal situation, you can see that these little vesicles or these little capsules are coming up to the front of the cell and they're doing their thing, okay? And you can see that here in still pictures. You can see if the lights are... I never asked, actually, can you see these pictures properly? Sorry. You can see that the little vesicles are getting up to the front of the cell here. However, if we mutate this capsule protein so it can't interact anymore with the enzyme, nothing happens up here. So our little vesicles cannot get up to the front of the cell. They just don't make it. So what does that mean in terms of the ability of the cell to move? What it means is that the lack of the movement of these proteins up to the front of the cell in the mutant, this is what happens when everything is normal. So if the vesicles are moving up, and you see these umbrella-like structures or these waves of actin, they're driving the cell forward. You can see them here in the still. When we mutate the RAB, the actin gets all spiky and basically all clumped up, and the cell isn't moving forward. And you can see this here. We do not get proper organization of this skeletal element that's pushing the cell forward. And I've just kind of summarized that in this cartoon. So the interaction of the enzyme and the vesicles, very important for driving this actin cytoskeleton forward in the positive direction. So this is, I've just described to you a couple of little experiments where we've decided, okay, RAC1 could be interesting in migration. We know that it does this, this, and this. So it's a candidate approach. So you're kind of making a good guess that it's probably going to be involved. But it would be nice sometimes to find proteins that you'd never guess would be involved in a process like migration. And these can be really insightful then and original in terms of designing drugs and all sorts of things. So we took this approach. What if we knock out more than 1,000 genes, just genes from a library, knock them out one by one in the T cells and see what happens? And how we knock them out, we use this technology called siRNA. And I don't really want to get too bogged down in the details, but siRNA is just basically when we have small little bits of RNA that bind to messenger RNA, which is going to be made into a protein, and degrades that messenger RNA so the cell can't express that protein anymore. That's what siRNA technology is in a nutshell. It got the Nobel Prize for Physiology and Medicine in 2006. So really fated as a very important technology. So we use this to ask the question, what happens if we, as I said, knock down genes one by one in the T cells, and what will happen? So we took the library of siRNAs, and one by one, we gave the cells an electric shock, not the cells one by one, but for every siRNA pool, we gave the cells an electric shock, delivered in an siRNA, and asked if they could migrate. And when you're doing such a big experiment, you're not going to physically sit in front of a microscope with 1,000 different experiments and do the analysis. We had a fabulous, and still have, not this one, but something very, very similar, technology called high content analysis or automated microscopy. And basically, the automated microscope does all the work. So you knock down your genes, you stimulate them to migrate, and you ask the microscope to make measurements on the cells. So you can make all sorts of measurements about the shape of the cell, the intensity of the fluorescence, whether, if you do it in live, whether it's moving or not. And just as well, we have that technology because we want to see what happens to the cytoskeleton from this standing start position. We all know that this is the optimum. This is what makes the cells move. Actin at the front and back, microtubules, train track, and this is what it looks like stained. When we knock out genes one by one, we see all sorts of different shapes and sizes. So the cells round up, they form star shapes. They can't, that cell looks like it's a super mover, but actually it doesn't move at all. These cells basically cannot move. So how do we quantify this? Basically, the automated microscope makes these measurements. So we can actually ask, what's the surface area of this cell? Where is the nucleus relative to the middle of the cell? It's in the middle here, but it's to one side here. The machine basically makes all sorts of measurements. So we can make then, for every treatment, we can make a profile for the cell. And this profile is basically telling us that the cell looks like this in this example. So this is actin here, and we get a big increase in actin. The nucleus is no longer to one side of the cell. So this is decreased. Green means decreased, red means increased. So these measurements are all made automatically. So when we do thousands of genes, then we can put or layer all those signatures for every single gene we looked at. And that's basically the experiment here in summary. All the signatures on top of one another and pick out what ones look the same, what ones look different, because that'll tell us about those genes and how those genes regulate the cell and how those families of genes regulate the cell. And we did find some really interesting things when we looked at that. We found a family of enzymes called casein kinase enzymes that regulate the migration, other single enzymes that also do. So it was a really big experiment that gave us loads of information that we're still actually working our way through. It's enough to keep you going for a long, long time. So just in the last few minutes, I want to tell you a little bit about the work that we've done in the context of T cells and disease, because this is ultimately why we are looking at these T cells and migrating T cells. And the first example is the hepatitis C virus. So we've known about hepatitis C for, probably more than 30 years. And clinically, there are very good drugs, curative drugs for hepatitis C. However, it is a huge problem because people end up with chronic hepatitis C being hard to diagnose. People are going around even with hepatitis C that hasn't been detected, particularly in third world countries where they don't have the money actually to do the relatively expensive diagnostic tests, and I guess maybe the relatively expensive drugs as well. Anyway, we're under a challenge, I think, to clear. The WHO want hepatitis C to be a thing of the past by 2030, but I think in 2020 there were 57 million people infected with hepatitis C and 300,000 deaths a year. So it still is a big problem. It's a virus. Our immune system, I told you way back at the beginning, is very powerful. It should be able to kill virally infected cells. But we all learned from COVID-19 and the SARS-CoV-2 virus that viruses are very clever, and they find out ways in which they can actually evade or avoid the human immune system. We learned about variants and mutations. We all became experts on viral infections. And hepatitis C is no different. It has multiple ways of avoiding the immune system. And one of the ways that it does that, we found out, coming back to our migrating cells and our polarized cells, we found that, and this is going back to way back, the beginning, PKC beta was right at the beginning. The PKC beta was actually necessary for the cells to be able to take that shape and migrate. If we knocked out beta, the cells stayed around. And lo and behold, if the envelope protein of the hepatitis C virus comes in contact with these T cells, the T cells stay around. They actually can't migrate. So the virus is stopping the cells from moving and causing the inflammation necessary to clear the virus. And when we saw that pattern, so PKC beta normally associated with the microtubule cytoskeleton, when we saw that pattern, we thought they look like lipid rafts. And these are specialized areas that are rich in cholesterol in the cell membrane. So we asked the question, is that what we're looking at? And yes, it is. So if we look in the resting cells, red is PKC beta and green lipid raft. If we treat the cells with the envelope protein, that becomes yellow around the edge. You can see that the envelope protein shunts this enzyme that's required for the cells to move into these lipid rafts. And that's how the virus, or one of the ways in which the virus subverts the immune system. Second example of migration and the disease, and I've already mentioned it, is the idea of inflammatory bowel disease and these gut homing T cells. So these T cells that get into the gut from the circulation, just as I described to you earlier, and cause inflammation. This is what happens in inflammatory bowel disease to give you these ulcers. And the thing is that when they get in there, they stick in there and they stay there and they cause tissue damage and these inflammatory sites. And we were looking at ulcerative colitis and Crohn's disease in a pediatric cohort. And we were looking at what we call an antimicrobial peptide. And that immediately has connotations of something that's protective. But we found that this antimicrobial peptide called beta-defensin 2 is overexpressed in the gut of children that have inflammatory bowel disease. So we asked, this protective peptide, does it have some kind of pathogenic effect as well? So put some of this beta-defensin into the bottom well of one of these assays and asked if it could attract these nasty gut-homing T cells to cross the barrier. And indeed they could. So while it's a bit of a double-edged sword, this antimicrobial peptide, which is protective in many instances, if it's overexpressed in the gut here, it is actually pro-inflammatory and pro-pathogenic and pulling in these gut-homing T cells. And finally, we'd be glad to know. This is a project that is quite new in the lab, and it is based on the idea of having T cells coming into the tissue and killing cancer cells. So we're looking at tumor infiltrating lymphocytes. And it's well known that tumors have cells, immune cells present. But some tumors have immune cells that don't actually promote this killing of the tumor. These are called cold tumors. And others then which will have lots of immune cells that are, anti-tumor, are called hot tumors. And in there, of course, will be T cells that can kill the tumor cell. And what also happens in these tumors is that the tumor releases soluble factors and the immune cells release soluble factors. And we could call it maybe a soup that's in there in the tumor. And we were asking, what is this soup doing to the T cells? So we put either tumor soup or normal tissue, soup derived from normal tissue, into these wells and look to see if they could invite in some T cells, or if T cells would come in. And indeed they do. They do come in response to this. But what we also, and this is preliminary data, but exciting, what we are seeing is that the soup that is derived from the tumor is actually killing the T cells that are going in. So we're seeing an increase in non-viable T cells. So it's one thing getting the T cells in there, but it's another to actually get them to carry out the function that they're supposed to. So the status quo, what do we want and why do we need to know all this, and why are we still the holy grail that we are looking for a situation where we get migration of T cells into the tissue, that they will be inflammatory until they clear the viral infection or the infection or the damage, and then that will resolve and won't cause disease. The same thing for cancer. We want the cells to come in, and we would like to have hot tumors rather than cold tumors, and functional cells rather than cells that are susceptible to death. A bit like the old having everything, on the status quo. The ATM is back in the wall and everything looks good. So thank you. Thank you. A lot of people here on this, PhD students, postdocs, colleagues, collaborators. It's been a wonderful journey in terms of research and, meeting lots of people. Thank you to the family, of course, my mom and dad and brother and sister. They've been, there from the very beginning, obviously, and a great support, and wouldn't be here without them. My husband and 2 daughters have been on the journey with me all the way along, and my rock have listened to me complain, have been there to celebrate the good times, but also, I guess, Shikra, who's online, and I hope she's still listening and not asleep, is having a great time in Canada, and Aisling is here and Peter is here. So thank you all very much, and thank you all. I'm honoured that you all came to listen to me this evening and celebrate this event with me. Thank you all so much.
What can I say, but thanks again for sharing not just a cross-section of what you've done, the people that you've worked with, but also about yourself and your journey, because it is, I think, a journey for everyone. I think given that Aideen has put so much work into that, we give her a break now. We don't usually take questions at this point, but we welcome you all to stay for the reception outside, and I'm sure you'll entertain any questions or photographs or whatever it is that people want to do at this stage. So thank you all very much.